Hello everyone, my name is AlanIzarraras and I am currently a PhD student in the International Laboratory for Human Genome Research in UNAM Juriquilla. And today I'll be presenting some work we have done on the Native American mitochondrial diversity in the Mexican population using ancient DNA. So the mitochondria is a single circular molecule of 16,000 base pairs that mostly codes for genes, but it also has a small non-coding region. It has some convenient characteristics, mainly for evolutionary and ancient DNA studies, such as that it is a single haploid molecule, meaning that it has no recombination. It can be found in high copy numbers in almost all cell types, and it is only inherited by our mothers. We now know that the Native American population diverged from ancient Siberians during the last glacial maximum, where they migrated from Siberia all the way to the American continent, establishing themselves as soon as 15 to 16,000 years ago. And with them, they carried some mitochondrial variation. Mainly, they carried mitochondrial haplogroups A2, B2, C1, and D1, although haplogroups D3H and X2A can also be found but are more rare. And some of these populations established themselves in Mesoamerica, giving rise to civilization. And because of the Spanish colonization, the present day Mexican population is mainly a mixture of Native American and Spanish ancestry, but also some African ancestry can be found due to the transatlantic slave trade. One of the main objectives of this study was to see if the ancient Native American mitochondrial diversity persisted in the present day Mexican population. And in order to do this, we sequenced and captured the full mitochondrial DNA from ancient samples, and we also gathered publicly available ancient and contemporary full mitochondrial DNA from published studies. So to test for genetic continuity, we constructed haplotype networks, but we also reconstructed the past female population size using a Bayesian skyline approach. So the samples were mainly taken from central and northern Mexico. So we have 2 from Sierra Tarahumara, 6 from Guanajuato, 8 from Querétaro, and 4 from Michoacán. In total, using 20 ancient samples for this analysis. Here we're looking at haplotype networks of haplogroups A2 and C1. In red circles, we have haplotypes from ancient samples, while the blue circles represent haplotypes from present-day individuals. And so, as you can see, no ancient haplotype seems to overlap with a present-day haplotype. And this pattern also persists in haplogroups B2 and D1, although they are not shown here. Only in haplogroup C1 did we find one example where an ancient and a present-day haplotype seem to overlap. We also reconstructed the past female population size, and so here we have population size in the y-axis and time in the x-axis. And so as soon as 4,000 years ago, we can see a demographic expansion followed by a decrease in population size that roughly coincides with the arrival of Spanish to Mesoamerica. And this is followed by a quick recovery. And so what this tells us is that although most Mexicans carry Native American mitochondrial haplotypes, some of these have been lost or at least made rare in the present-day population. Also, the Spanish colonization that took place about 500 years ago had a noticeable impact on mitochondrial diversity and the female population size. So thank you all very much for listening. Thank you to everyone at the Population and Evolutionary Genomics Lab Thank you at everyone for Lavis for the computational infrastructure. And of course, a special thanks to all of our collaborators. Thank you.